In this video, we will discuss about the nutrient supply in the cancerous cells. More specifically, we will talk about the pancreatic cancer cells, how the nutrient supply has been maintained by the pancreatic cancer cells. And there are two major ways by which these cancerous cells are able to maintain their nutrient supply, that is autophagy and macropinocytosis. So let us first understand how the normal cells are able to maintain their nutrient supply. One of the way by which cells maintain their nutrient supply is via glucose and glutamine. So this glutamine will go and convert it into the glutamate with the help of enzyme, and after that it will form this alpha Kg that helps in the TCA cycle. And simultaneously this glucose will also ultimately come and help into this TCA cycle. However, in the case of this pancreatic cancer, this pancreatic cancer, this GOT1 is usually expressed, and this GLUT is down expressed. Because of this, the glutamate will be converted into this glutamate will be followed by this pathways instead of going to this alpha Kg pathway. This is alpha Kg formation will be reduced, and by going through this pathway, that is with the help of enzyme GOT1, they will form NADPH. And however, most of this NADPH is being used by this pathways, that is used by most of the NADPH will be used by the further pathways. So in order to reduce this NADPH supply to the normal cell metabolism function, that signaling pathways are been down regulated in the case of pancreatic cancer. For example, this in this figure, the examples are not shown here, the where the NADPH pathways will be used, but it is generally used for the normal cell metabolism functioning. So that is downregulated in the case of pancreatic cancer. The other things that happens, so this is in general what happens in the case of pancreatic cancer. Now let us understand how this autophagy and macropinocytosis plays a role. So this autophagy means the recycling the internal uh, component of the cells, and so that those amino acid can be used by the cells for its uncontrolled growth rate and proliferation. So this is just an example that is shown here. And this, in the case of macropinocytosis, what happens is that it will take up the extracellular components that are surrounding the cells, and that will be recycled by the lysosomes and use those amino acids for its uh, tumor cell for the tumor cell progression. So and how this mTOR plays a role in this case is that when there is a autophagosome or when there is a less supply of the nutrients within the cells, it will trigger it will this inhibit this mTOR genes that are responsible for mTOR, and because of this, what will happen is that this early endosome and the lysosomes will be able to fuse together and will provide sufficient energy for the tumor cell survival. However, in the case of macropinocytosis, what happens is that since the nutrient supply is high in the case of macropinocytosis, so in this case mTOR, the genes that are responsible for mTORs will be upregulated, and that will prevent the fusion of autophagosome and lysosome, and thus the proteins or thus the recycling of the amino acid will not be diverted towards the normal normal survival of the cells, whereas it will be diverted towards the where it will divert it towards the uncontrolled growth rate and proliferation of the cells. So this is just a mTOR function in the case of autophagy or macropinocytosis, or we can understand in the case of this nutrient deprivation within the cells, or this nutrient excess within the cells. So mTOR is upper mTOR is we can say down regulated or inhibited when the nutrient supply is low, and mTOR is upregulated when the nutrient supply is high. And one more thing that will happen that helps in this extracellular components to have more nutrient supply. And for example, this alanine is being used by the cells as it is as a replacement of the glucose in most of the cases. And how this lactate helps in this nutrient supply formation in the cellular matrix is that this tumor cells, since this its intercellular metabolism pathway has been changed, it will secrete more lactate in the more lactate in the cellular matrix, that will decrease the pH of tumor, and that will lead to the dissolve of the fibrosis that surrounds this tumor cells. And the tumor microenvironment will be exposed to a will be exposed and increased towards containing these oxygenated cells, and thus those oxygens can then be utilized by using macropinosynthesis, uh, and thereafter the recycled amino acid from macropinosynthesis can be utilized for the tumor cells growth and progression. So that's how this autophagy and macropinocytosis works in the case of pancreatic cancer cells. If you need to understand in detail about how this autophagy and macropinocytosis are correlated, and what are the individual factors that are responsible for autophagy and macropinosynthesis, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.